Omega-3 fatty acids, particularly the long-chain varieties EPA, eicosapentaenoic acid, and DHA, docosahexaenoic acid, found in fatty fish and marine oils, have long been recognized for their cardiovascular and neuroprotective benefits. In recent years, they have also entered the spotlight for their potential role in slowing cellular aging, specifically in preserving telomere length. While findings in this area have been somewhat mixed, early clinical trials and mechanistic studies have provided a compelling rationale for omega-3s as a gentle, dietary-based strategy to support telomere integrity through their effects on inflammation, oxidative stress, and lipid metabolism. One of the core pathways through which omega-3 fatty acids may influence telomere dynamics is via their well-established anti-inflammatory properties. Chronic, low-grade inflammation accelerates the rate of telomere shortening, contributing to early cellular senescence and age-related diseases. Omega-3s modulate the body's inflammatory response by altering cell membrane composition and reducing the synthesis of pro-inflammatory eicosanoids, while simultaneously increasing the production of specialized pro-resolving mediators like resolvins and protectins. This anti-inflammatory shift is believed to create a more favorable biochemical environment for telomere maintenance, particularly in immune cells such as leukocytes, which are commonly used in telomere studies. In addition to dampening inflammation, omega-3s are known to reduce oxidative stress, another major contributor to telomere attrition. DHA and EPA can enhance antioxidant enzyme expression and help protect cell membranes and DNA from oxidative damage caused by free radicals. Since telomeres are especially vulnerable to oxidative damage due to their high guanine content, any intervention that lowers systemic oxidative burden has the potential to slow their rate of erosion. In early human trials, individuals with higher blood levels of omega-3 fatty acids were found to have longer telomeres and slower telomere shortening over time, particularly when accompanied by reduced levels of oxidative markers and inflammation. However, more recent studies have introduced nuance into this picture. Not all trials have replicated these positive findings, and discrepancies often seem to relate to differences in dosage, study duration, baseline omega-3 status, or the population studied. For example, some shorter-term trials using low doses or studying already healthy individuals have failed to show statistically significant changes in telomere length. These results suggest that omega-3's effects on telomeres may be subtle and require prolonged supplementation to manifest, especially in individuals without elevated baseline inflammation or oxidative stress. Alternatively, the benefits may be more easily detected in populations with higher biological stress levels, such as those with metabolic syndrome, obesity, or chronic disease. Still, the mechanistic plausibility and safety profile of omega-3 supplementation make it an attractive candidate for long-term health support, especially when incorporated as part of a broader anti-aging lifestyle. Beyond telomeres, omega-3s support brain structure, cardiovascular resilience, eye health, and emotional well-being, all of which intersect with cellular vitality and aging. When combined with other telomere protective behaviors such as exercise, sleep optimization, and stress reduction, omega-3s can play a meaningful supportive role in promoting cellular longevity. Furthermore, emerging research is beginning to explore the effects of personalized omega-3 dosing, taking into account factors like omega-3 index, a blood marker of EPA plus DHA levels, genetic polymorphisms, and dietary background. This individualized approach may help resolve some of the inconsistencies in the research and maximize therapeutic outcomes. It's also worth noting that omega-3s derived from food sources such as salmon, sardines, anchovies, and flax seeds are often accompanied by synergistic nutrients that enhance their bioavailability and effect, offering an additional advantage over isolated supplements in some contexts. In summary, while omega-3 fatty acids are not a guaranteed method for lengthening telomeres, their consistent ability to reduce inflammation and oxidative stress makes them a biologically plausible and practically achievable component of a telomere-friendly diet. The mixed findings in the literature do not diminish their value, but rather underscore the importance of context. Dosage, duration, and individual health status all matter. As part of a long-term strategy, omega-3s offer more than just heart and brain support. They may help protect the very tips of our chromosomes, contributing to a healthier, more resilient aging process.